Hello and welcome to Lecture 3 on the TCA cycle. What we're going to do in this is we're going to start off with talking about energetic consequences of TCA. Or really, what are our outcomes? So, for the TCA cycle, per unit of acetate oxidized, in one cycle of TCA, we get 2 molecules of CO2, 1 molecule of ATP, 4 reduced enzymes, 3 of which are NADH, and 1 of which is FADH2. And so what this looks like in terms of the overall balance reaction is acetyl-CoA plus, as an input, 3 NAD plus an FAD plus ADP plus inorganic phosphate plus 2 water yield 2 equivalents of CO2, 3 NADH, 3 protons, 1 FADH2, 1 ATP, and a coenzyme A. So the delta G for one pass of the cycle is approximately negative 40 kilojoules per mole. Which is overall very favorable. So glucose, going back to glucose, metabolized via glycolysis produces 2 molecules of pyruvate. So 1 glucose will yield 2 pyruvate, and therefore 2 molecules of acetyl-CoA. Which can enter TCA cycle directly. And if you combine glycolysis and TCA, you can just multiply all of these outcomes by 2 per unit glucose. But that's for TCA only. If we actually combine the balanced equations for glycolysis and TCA, it looks like this. We have per glucose and 2 water, 10 NAD plus 2 FAD, 4 ADP, 4 inorganic phosphate. As an outcome, we get 6 CO2, 10 NADH, 2 FADH2, 10 protons, should have put those right after the NADH, and 4 ATP. Now, note that all 6 carbons of glucose, because glucose has 6 carbons, are liberated as CO2, and a total of 4 molecules of ATP are formed via substrate level phosphorylations. The 12 reduced coenzymes produced up to this point can eventually produce as many as 34 molecules of ATP when they enter the electron transport and oxidative phosphorylation pathways, which we'll see in a following chapter. So let me just highlight that real fast. Note that these NADH and FADH2 will enter electron transport, and we would expect these to contribute as much as, depending on the degree of efficiency in the system, as many as 34 more ATP. So per glucose, we would expect to see about 38 combined ATP produced, with complete oxidation to CO2. The way this actually breaks down, if we look at the NADH real fast, this is all approximates, but you get about 3 ATP per NADH, and you get about 2, these are approximately, ATP per FADH2 when these enter electron transport plus oxidative phosphorylation. Now, these numbers are always subject to revision. You see over the course of textbooks these numbers drift a little bit, but ballpark 3 and 2 is a reasonable estimation. You may think, why 3, why 2? Well, note that the rationale for FADH2 forming to capture the energy in the formation of a double bond was because there's less energy available in that transformation than in the oxidations that were taking place prior. And this lower amount of metabolic energy captured is reflected in the fact that there's less metabolic energy available in this reducing equivalent to form ATP. So that's why we're seeing these different amounts of ATP formed per reducing equivalent between NADH and FADH2. So now that we've reviewed energetic consequences, let's go ahead and make a little space here, erase all this stuff. And we're going to go into alternative roles or functions of the TCA cycle in addition to energetics. So until now, we've primarily viewed the TCA cycle as a catabolic process because it oxidizes acetate to units of carbon dioxide, converts the liberated energy into ATP and reducing coenzymes, which then go on to make more ATP. So it's a classic catabolic process. So the TCA cycle is, after all, the end point for breakdown of food materials, at least in terms of carbon turnover. However, there are a number of roles of the TCA cycle associated with biosynthetic processes. So first off, let's take a look, quickly let me sketch the TCA cycle here. So we have pyruvate, and we have acetyl-CoA, and then this, we have oxaloacetate combining with acetyl-CoA forming citrate, which then goes to isocitrate, which then goes to alpha-ketoglutarate, which then goes to succinyl-CoA, which then goes to succinate, then fumarate, malate, and back to oxaloacetate. So, I know, we literally just looked at this, Dr. McGill, right, but I just wanted to give everyone an overview of all of the various, the classic structure of this, so we can start looking at the other roles of these metabolites, because these are primary metabolites. And it's not this neat linear circle that just goes round and round and round in a circle, generating reducing equivalents and reduced coenzymes and ATP. Another really important role is how these metabolites feed and replenish other metabolic cycles. So, example, alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate are all precursors to important cellular species. So, firstly, a transamination reaction converts alpha-ketoglutarate directly to glutamate, which can then serve as a versatile precursor for proline, arginine, and glutamine. And we'll take a look at that down the road. But what's this look like? We can shuttle alpha-ketoglutarate which will become glutamate, which can become, let me make a little bit of space here, proline or glutamine, which then feeds into purine nucleotides, actually. And then glutamate can go on to a number of other things, including citrulline and arginine. And then we have succinyl-CoA, which provides most of the carbon atoms for porphyrin rings. So, make a little bit of space here. Succinyl-CoA can make porphyrin rings. I should probably have more arrows in here, a sequence of arrows, because it's not super direct, but that is one of the terminal fates of that. Following that, oxaloacetate can do a lot of different things. It can be transaminated to produce aspartate, and then aspartic acid itself is a precursor to the pyrimidine nucleotides. In addition, it's also a key precursor for the synthesis of asparagine, methionine, lysine, threonine, isoleucine. So let's take a look at that. Oxaloacetate can actually feed in here, and actually fumarate can as well. Take a little room on this side. And then this can form asparagine. We can get all of our pyrimidines. And then I'm going to draw a couple arrows here because it's a diverse pathway, but you can get your threonine, isoleucine, methionine, and lysine. So tons of amino acid pathways and nucleotide pathways coming out of this, apparently, as well. In addition to this, oxaloacetate can also be decarboxylated to yield phosphoenolpyruvate, which is a key element of several pathways. In particular, it helps drive gluconeogenesis, which we'll see in chapter 22 is a pathway that synthesizes new glucose and many other carbohydrates as well. So I'm going to draw from oxaloacetate up here. We need to make some more space again. Decarboxylation. And we can get phosphoenolpyruvate, or PEP. And then from here, we can go on to making new carbohydrates. We can also, phosphoenolpyruvate's involved in the formation of tryptophan and tyrosine. And all kinds of things going on here. So, let's see. Oh, and in addition to this, if we look at alternative roles of acetyl-CoA, acetyl-CoA is super important for lipid synthesis. So acetyl-CoA goes into malonyl-CoA, which is for your fatty acids. It also is involved in your formation of steroids, which are classes of lipids. And let's see, is there anything else here? I think that's a pretty good summary. So we have a lot of different functions already for TCA cycle. Let me actually highlight this. So within this core, which is our TCA cycle, if I zoom back in a little bit, you can see a bunch of different roles, which include porphyrin rings with our porphyrins, our purines, our pyrimidines, tons of different amino acid syntheses, our lipid and steroid syntheses, and carbohydrate synthesis. So we've just covered all of our macromolecules. and tie them all back to our primary metabolism. So one last thing, which will be really important in our transition to discussing anaplerotic or filling up reactions, is a role for citrate. Let me zoom back in here. So citrate can be exported from the mitochondria and then broken down by an enzyme called ATP citrate lyase, which yields oxaloacetate and acetyl-CoA. So citrate can leave the mitochondria and make you oxaloacetate and acetyl-CoA, basically going the other direction from its formation, which makes sense as just part of a cleavage reaction. And of course, acetyl-CoA can then be used to make more fatty acids or steroids. Oxaloacetate produced in this specific reaction, because it's no longer in the mitochondria, is rapidly reduced to malate, going the opposite direction. Let me grab a different color here. Remembering this is not mitochondrial because it's been exported. And this can be processed one of two different ways. It can be transported back into the mitochondria where it's reoxidized to oxaloacetate because oxaloacetate is maintained at a very low level. Or, my favorite, it may be oxidatively decarboxylated to pyruvate by malic enzyme. So this malate can be converted into pyruvate with malic enzyme. And what this does, and then following that, it's subsequently taken up. So what this does is this cycle permits citrate to provide acetyl-CoA for biosynthetic processes with the return of malate or pyruvate byproducts to the mitochondria. Very, very slick way, if we're low on acetyl-CoA, to re-engage in lipid metabolism directly. Another role of this is really interesting, and it's, well, actually, let me return to this in just a second. I'm going to talk about anaplerotic reactions, and then we're going to come back to malic enzyme, because I think that this warrants a little bit of an elaboration. So I'm going to make a little bit of room here. Just kind of scroll up out of the way. Let's talk about anaplerotic reactions. Well, an anaplerotic reaction literally means a filling up reaction. And what this is it's in a sort of reciprocal arrangement. Cells also feed many intermediates back into the TCA cycle from other reactions. So we just saw how TCA can supply the cell with all kinds of metabolites. Well, the cell can also push back into the TCA cycle or citric acid cycle without coming in from the top with acetyl-CoA. And this helps replenish zones which may be low for any variety of reasons. And there's three reactions that we're probably going to pay attention to here. Let me go back to black. We have phospholynyl pyruvate carboxylase. We have pyruvate carboxylase. And we have malic enzyme. And what these reactions look like are, for pyruvate carboxylase, well, we'll start with phospholynyl pyruvate carboxylase. You have phospholynyl pyruvate, which has your carbon double bound here and our phosphate. And what this does is it converts phospholynyl pyruvate to oxaloacetate. And to do this requires the addition of water and CO2. and we actually get a inorganic phosphate out from the loss of the phosphate here. So what's conserved in this reaction? Well, you can see that the CO2 is being added at this position using the double bond as a target for that specific addition. In addition to that, we're also adding water. So let me change back to black here. I probably didn't leave myself enough room, so I'm going to draw a little bit staggered here. That was phosphoryl pyruvate carboxylase, and then we have pyruvate carboxylase. Well, tripping on myself here. Pyruvate carboxylase starts with pyruvate. Which is remarkably similar to phosphatal pyruvate, except that it's in the keto form and there's no phosphate. So this also converts to oxaloacetate. So both of these systems are able to replenish oxaloacetate via phosphatal pyruvate and pyruvate respectively. Now, pyruvate carboxylase doesn't just require CO2, it also requires the addition of ATP because it's a much more stable compound. So this is a very expensive process energetically, but sometimes it's worth spending that money if you need to fill up. And likewise, the equivalent of CO2 being added. Now, finally, we've got malic enzyme. And malic enzyme is not producing oxaloacetate. It's taking pyruvate and converting it into malate. Or it can convert malate to pyruvate. But I'm going to draw it pyruvate to malate first. So we have our pyruvate. And it's going to go to malate with the addition of a CO2. Now, what's unique about this one, and one of the reasons I like this reaction so much, is because, as drawn, this actually consumes an equivalent of NADPH as a reducing equivalent to make an NADP+. And we haven't seen a lot of NADPH because we've been looking at reactions which are associated with energetics, and catabolic reactions are typically NADH, or exclusively NADH. NADPH is an anabolic reducing coenzyme, and it's used primarily for lipid biosynthesis, because something has to supply the high energy electrons for the carbon-carbon forming bonds, carbon-carbon bond forming reactions that take place in lipid synthesis. Now, another thing that happens here, is we can also see the reverse reaction. So remember, let me scroll up here. I had an arrow specifically showing malate going to pyruvate via malic enzyme. And that's because oxaloacetate produced in the reaction with ATP citrate lyase had those two fates, right? It could be transported in the mitochondria or was reoxidized to oxaloacetate, or the malate can be oxidatively decarboxylase to pyruvate by malic enzyme, subsequently being retaken up in the mitochondria as pyruvate. And so not only can the reaction go as drawn, but it can go in reverse. And the reason that's important is because malate can also produce pyruvate, and by doing so, generate NADPH. What we'll see in the future with lipid biosynthesis is that this is a very expensive, making lipids is very expensive. Making carbon-carbon bonds costs a lot of energy, and we spend a lot of energy making NADPH. So the other reason we really need NADPH in addition to lipid biosynthesis is for redox defense. Because all of our redox defense systems, I'm going to go on a little segue here, rely on enzymes which actively scavenge free radicals. And this includes superoxide dismutase, or SOD, catalase, enzymes along these lines. So these enzymes are currently extremely important for scavenging free radicals. In addition to this, they don't have an infinite capacity to execute this function. They need to be replenished as well. And the way that they're primarily driven is through glutathione, GSH. Now, glutathione doesn't have infinite ability to act as a reducing agent, and when it's consumed, it exists as a dimer, glutathione disulfide, where we have 2 units of GS bound to one another through the sulfurs. To push it back and remake active glutathione, we need NADPH. So ultimately, our ability to scavenge free radicals or redox defense in general is mediated by glutathione. And glutathione is also very important because it controls a lot of redox-sensitive transcription factors, which includes genetic regulation of these systems. But ultimately it comes down to, do we have NADPH? So there are periods of time where we have competing needs for limited pools of NADPH, and the ability to rapidly produce NADPH metabolically is very, very important, specifically when you're dealing with infection, when you're dealing with some kind of specific acute redox insult. And under those circumstances, we can literally scavenge malate from the TCA cycle, export it from the mitochondria into the cytosol, use malic enzyme, convert to pyruvate, produce NADPH, and help drive our essential physiological functions via this particular anaplerotic reaction. So anaplerosis can go in, but it can also be another mechanism of drawing some of these primary metabolites back out again. Okay, and now that we're all zoomed back out, looking at the big picture, what's final for this chapter? Let's discuss overall how the TCA cycle is regulated, because we need to understand how all of our energetics help reciprocally control this cycle. So let me erase a little bit of this, make some space, and then we'll zoom back in. And I want you to keep your eye on, well, I just erased it, pyruvate carboxylase. Remember that pyruvate carboxylase directly converts pyruvate to oxaloacetate as one of the anaplerotic reactions that we just erased. So I'm going to get rid of that. I'm going to get rid of some of these things over here. Get rid of the lipids and carbohydrates. Now we're going to zoom in and have a relatively fresh TCA cycle. Maybe get rid of some of these peripheral lines. instead of rewriting the whole thing, it may have been faster just to rewrite the whole thing. We'll see. Close enough. All right, we're going to zoom in some more. And then I'm going to make one more line with pyruvate able to go directly down to oxaloacetate via pyruvate carboxylase. So we have a couple important enzymes here. We have, let me see, pyruvate dehydrogenase, which is managing the conversion of pyruvate to acetyl-CoA. We have citrate synthase, which is facilitating the combination of acetyl-CoA and oxaloacetate. We've got isocitrate dehydrogenase, which is a conversion of isocitrate to alpha-ketoglutarate via the loss of CO2. So I'm not going to draw the loss of CO2. We know that from earlier. We also have alpha-ketoglutarate dehydrogenase for the following step. And these, and I'm going to include this up here, pyruvate carboxylase. These are all of the enzymes which are really critical for regulation of TCA. 1, 2, 3, 4, 5. So let's take a look at these regulations. In terms of inhibitors, let's start with pyruvate dehydrogenase. We have, I'm going to go minus for inhibitors, plus for activators. We have acetyl-CoA, NADH, ATP. And as an activator, we have NAD+, and we have coenzyme A. For citrate synthase, all inhibitors. ATP, NADH, succinyl-CoA. Make a little space here. For isocitrate dehydrogenase, inhibitors include ATP and NADH. Activators include ADP. At alpha-ketoglutarate dehydrogenase, inhibitors are NADH, succinyl-CoA. Rewrite that. And activators include AMP. It doesn't hurt to put negatives next to everything that's negative and positives next to everything that's positive. It helps keep it clean. And then pyruvate carboxylase is activated by acetyl-CoA. And that makes sense because we have a feedback need driven in to this allosteric regulation. But that's hard for me to erase now because I can't selectively erase reds. So let me zoom back out. We'll make it in center here. And this is a good tidy picture of how TCA is regulated in terms of specifics and in general. In general, you should be able to rationalize why negative inhibitors act the way they do, why positive inhibitors act the way they do. There should be a clear biological rationale for their function. And I think, check my notes, that's going to be it for the chapter on TCA cycle. So I'm going to get this compiled, loaded up to the channel, and I'll be talking to you all next about electron transport and oxidative phosphorylation. So see you all soon.